Assalamu alaikum. So in this lecture we'll be talking about the cell cycle. So the cell cycle is basically a process by which cells divide into two cells. Now the time it takes for a cell to enter this process and finish it is about 24 hours for eukaryotic cells. Now it's different between cell to cell type, okay, but usually for a eukaryotic human cell, at least in the lab in culture, it takes about 24 hours. It takes about 23 hours before going through the separation of chromosomes and cell division, and this is called mitosis. Okay, it takes about only an hour for mitosis to take place. Now, it takes yeast cells about an hour and a half to divide. It takes bacterial cells about 40 minutes to divide. Okay, so it's different between cell type to cell type. Now, in a zygote, okay, so here's the process basically. Cells go through a phase known as G1 phase. G stands for growth. So cells would continually grow and accumulate metabolites and molecules that are needed for the next phase, which is the DNA synthesis phase, also known as S for synthesis. Now, after going through the S phase, they go through a G2 phase, whereby they keep on growing, okay, preparing themselves for the next phase, which is the M phase or the mitotic phase. Now, during the G2 phase, again they synthesize proteins that are needed for the mitotic phase. Now, so now zygotes, by the way, do not go through G1 and G2 phases. They already have what they need for the process for DNA to be synthesized and cells to divide. Now, some cells do not, once they are differentiated, and they do not divide. Once they are formed, that's it, they do not divide, and these are including nerve cells. Now, these nerve cells, they stay in a phase known as the G0 or G0 phase, whereby they are quiescent. They are just quiet. They only do metabolism. They do not grow. So these are the phases again. G1 is for growth, metabolism. Cells are diploid. Now, in the DNA synthesis phase, cells are between 2 to 4N because now DNA is synthesized. Now, in the G2 phase, the cells have 4N. They have double the amount of chromosomes. Now, in the M phase, they segregate, so they start with 4N and they end up with 2N in terms of chromosomes, chromosomal copies. Now, the cell cycle must be strictly regulated. Okay, so before they jump from one phase to the other, they have to go through a restriction point, or they have to go through a point whereby there is no point of return. Now, this point is known as the restriction point, and it exists in the G1 phase. So right before going through the final stages of the G1 phase, synthesizing the proteins that are needed for DNA synthesis, they have to go through a checkpoint. Okay. Now, this restriction point is overcome in the presence of growth factors. So in the presence of growth factors, cells can jump over the restriction point. Otherwise, if they do not, they would stay in the G0 phase. Okay. Now, some cells, some cells are always in the G1 phase, like nerve cells. Now, other cells are in the G1 phase, but they can they can enter the G1 phase, getting themselves ready for the S phase, such as skin fibroblasts. Okay, now these are arrested in the G1 phase until they are stimulated by platelet-derived growth factor, which is a growth factor. So what happens is, whenever there is an injury in the tissue and these fibroblast cells are damaged, they would exit the G0 or G0 phase and they enter the G1 phase in order to proliferate and compensate the loss of cells. And then once they divide to a certain number, that is, it's always controlled, they enter the G1 phase. So there are a number of checkpoints, by the way. Okay, so these checkpoints, cells sort of like scan their readiness to make sure that before they go into the next phase, they are ready. So for example, there are checkpoints for DNA damage. So there's one in the G1 phase, there's one in the S phase, and there's one before entering the M phase. In the M phase, there is a checkpoint that is specific for spindle assembly, that is the preparation of microtubules that are necessary for the separation of chromosomes into both ends of the cells. So right at the end of the G1 phase and right before DNA synthesis, cells try to make sure that DNA is not damaged, that there are no mismatches, that everything is fine before copying DNA. During the S phase, again, cells scan the DNA that is copied, making sure that it does not contain any mismatches. And right before cell division, again, cells make the final check of DNA, making sure that there are no mutations or DNA damage. Now, how is the cell cycle regulated? They are regulated by specific proteins. And there are three classes of proteins that regulate the entry of cells into the S phase or into any one of the phases that we've talked about. Now, these three classes of proteins are known as cyclins, cyclin-dependent kinases, and CDK inhibitors or cyclin-dependent kinase inhibitors. Now, these cyclins, by the way, they are proteins that accumulate throughout the interphase, and this is something that I forgot to mention, that from the beginning of G1 phase all the way to the end of the G2 phase, it's known as the interphase. So these cyclin proteins, they accumulate throughout the interphase and they are rapidly degraded at the end of mitosis. So what happens is that you have these cyclin proteins that keep on cycling in terms of levels. That's why they're called cyclins. So their level increases, and then at the end of mitosis, they are degraded. Okay, they increase, and then they get degraded, and so on. Now, cyclins are important for the activity of cyclin-dependent kinases. So what you have in here is a dimer, a protein dimer that was the first protein that was identified, and it's called the maturation promoting factor. This maturation promoting factor was found to be critical for cell division. So it's a dimer that is made of a cyclin known as cyclin B associated with a cyclin-dependent kinase known as Cdk4. So the catalytic activity of Cdk4 is dependent on the presence of cyclin B. Okay, so this Cdk4 and cyclin B are needed in the G2 phase right here. Okay, so let's look into a specific example of how cell cycle is regulated. In this example, we'll be talking about Cdk1 complexing with cyclin B. So we'll talk about the complex of CDK1 and cyclin B, which allows for cells to enter the M phase and continue till the end. Now what happens is, first you have the synthesis of cyclin B. Now this cyclin B then complexes with CDK1. Now this complex here allows for the phosphorylation of CDK1. CDK1 is phosphorylated at 3 positions, 3 amino acids, which are threonine 14, that is amino acid number 14, which happens to be threonine, tyrosine 15, and threonine 161. Now this complex is not active yet. Okay. In order for it to be active, these 2 phosphates on threonine 15, threonine 14 and tyrosine 15 must be removed. So the protein must undergo dephosphorylation. Now this complex, by the way, is the same as the maturation promoting complex or factor. Now what happens then is now since these 2 positions are dephosphorylated, now CDK1 is active. CDK1, it's a kinase. It starts to phosphorylate many proteins, allowing for cells to enter into the M phase. Now the protein CDK1 then or the cyclin B specifically undergoes degradation, resulting in inactivation of CDK1. This is followed by dephosphorylation of the activating position right here, that is threonine 161. So this phosphorylated site makes the protein active, by the way, and these 2 positions, when phosphorylated, they make the protein inactive. Now what happens then is that so CDK1 is dephosphorylated, and the cycle starts all over again by synthesizing cyclin B, complexing with CDK1, and so on. Okay, so it's all regulated. Now, so we have different complexes, right, between CDKs and cyclins. So for example, the complex of CDK4 with cyclin D or CDK6 with cyclin D would allow for cells to go through the G1 phase. The complex of CDK2 with cyclin E would allow cells to enter the S phase and finish it, to get through the S phase, that is. The complex of CDK2 with cyclin A would allow cells to complete the S phase until it get near the M phase. CDK1, cyclin A, would allow cells to finish the G2 phase and enter the M phase. And then we have CDK1, cyclin B, would allow cells to get into the M phase and complete it. Okay, so notice that different complexes have different purposes. Okay, now, so there are different modes of regulation of CDK activity. 1, they must associate with cyclins in order for them to be active. That's 1. 2, they are activated by phosphorylation of threonine 161. Okay, so there are specific kinases known as CDK activating kinases that phosphorylate these positions right here. Now, phosphorylation of threonine 14 and threonine 15 would inactivate the protein, and they must be dephosphorylated. Finally, they associate with CDK inhibitors. Okay, so each one of them has a specific inhibitor, and their dissociation from that inhibitor or the degradation of the inhibitors would allow for activation of the cyclin-dependent kinases. Now the cell cycle is linked to cell signaling and growth factors. So the presence of growth factors would indicate that there are enough nutrients, the environment around cells is promising for cells to divide, and so on. One of those signaling pathways is the RAS pathway that we talked about before. Now, the presence of growth factors would induce signaling of the RAS pathway. Now, what happens is that, if you remember, you have phosphorylation of ERK at the end. ERK goes into the nucleus. It binds to the regulatory sites on promoters of certain genes. One of these genes is cyclin D. Increasing the level of cyclin D would make it allow it to associate with Cdk4 as well as Cdk6, and facilitating the transition from the G1 phase to the S phase. And it just happens, by the way, that defects in cyclin D would allow for cells to continually grow and divide, a characteristic of cancer cells. In fact, I'm involved in a project with one researcher and 5 students. We're looking at the expression of cyclin D as a diagnostic marker for a type of brain tumor known as ependymoma. In addition, by the way, these CDK or CDKs have been targeted by certain drugs as a strategy of treating cancer, by the way. Now, any defects in the inhibitors of CDK would allow for these cyclin-dependent kinases to be active, and mutations in these inhibitors would allow for, again, for loss of control of cell proliferation. One of those proteins that are found to be associated with cancer is retinoblastoma protein. So retinoblastoma is actually a tumor suppressor gene. It prevents the formation of tumors by inhibiting the cell cycle, progression through the cell cycle, that is. So what is retinoblastoma? Retinoblastoma is a type of cancer that affects children primarily, whereby they develop tumors in their eyes. And it is related to mutations in the retinoblastoma protein. Now, the retinoblastoma protein normally forms a complex with elongation factor 2, preventing transcription, okay, and preventing cells from dividing. Now, what happens is that when Cdk4 or Cdk6, when they are associated with cyclin D, now the kinase is active. It phosphorylates retinoblastoma, releasing it from the elongation factor 2 and allowing for transcription to proceed and for cells to divide. One of the genes that are activated is cyclin E. Cyclin E is the cyclin that is necessary for cells to go through the S phase, that is the following phase. Because Cdk4 and 6, when complex with cyclin D, they allow for cells to go through the G1 phase. So here we have cyclin D associated with cyclin-dependent kinases 4 or 6. They allow cells to go through the G1 phase. Then we have the cyclin E associated with CDK2, allowing cells to go through to enter the S phase and go through the initial stages of the S phase. So the elongation factor 2 then induces synthesis of cyclin E. But there are other things that also happen to allow cells to go through the cell cycle. One of them is that the inhibitor of cyclin-dependent kinase 2, which is known as P27, is relieved, or it's inhibited. And it is inhibited by a number of mechanisms. One of them is that in the presence of a growth factor, that induces signaling through the RAS pathway as well as the PI3 kinase AKT pathway. that reduces both the transcription and translation of P27. So lowering the levels of P27, allowing for CDK2 to be more free, and once it is more free, it can induce cell cycle progression. Now, activated cyclin-dependent kinase 2 induces degradation of P27, lowering the level of P27 even further. So not only that you have reduced synthesis, you also have more degradation. Now what happens is that once you have P27 inhibited, that would activate cyclin E, cyclin-dependent kinase cyclin E complex would be more active, phosphorylating many proteins, including cyclin E itself. Okay, and when cyclin E is phosphorylated, it becomes more stable. Okay, now this high level of cyclin and CDK2 it would then be maintained, allowing for cells to go through the cell cycle, the S phase as well as the G2 phase. Remember, if you look at it, cyclin-dependent kinase 2 is important for both the S phase as well as the G2 phase. Now, one important protein in the development of cancer is known as P53. P53 is a tumor suppressor gene. Its level must be high in order to prevent cells from growing and proliferating without control. And mutations in P53 have been found to be associated with development of cancer. Now P53, by the way, is a sensor for cell damage, for DNA damage. So if DNA is damaged, P53 would be activating, preventing cells from going through the cell cycle until DNA is repaired. Now this P53, by the way, is activated by two proteins. One of them is known as ATM and the other one is known as CHK. Now ATM is a sensor for DNA damage. So whenever there is DNA damage, ATM is activated. It phosphorylates P53 either directly or indirectly by activating CHK2. Now phosphorylated P53 can then sit on DNA, preventing increasing the expression of P21, which is an inhibitor of cyclin E and CDK2 complex. Okay, so preventing cells from progressing through the S phase. Now, so something else that is important, one target of cyclin B CDK1 complex, which is necessary for entry of cells through the M phase, is that what they do is that they phosphorylate proteins. Among the proteins that are phosphorylated is the nuclear envelope, the lamin proteins. Remember when we talked about the lecture, in the nucleus lecture, when we talked about regulation of lamins is by phosphorylation, and phosphorylation of lamins results in their dissociation. Okay, so this is one step by which cells must do to get into the M phase, that is degradation of the nuclear matrix and the nuclear envelope eventually, so that chromosomes would be released into the cytoplasm, putting them or making them ready to be separated through mitosis.